In this work, we want to use genetic and clinical data to help predict entire anatomical brain MRIs. In particular, we assume that we have a single or baseline MRI scan from a subject, say at 50 years old, and we want to use this patient's genetic and clinical information to predict their MRI scan at, say, 60 years old. We'll first talk about a simplified problem of predicting a single scalar number, or phenotype, and after I'll show how to predict an entire MRI follow-up scan. We assume we have a longitudinal population of subjects, and we have measurements for all of them for some phenotype, such as hippocampal volume. Then we observe only the baseline or first measurement of a new subject, and we're interested in predicting the phenotype for that subject at a later time. We model this change in phenotype as a linear regression with respect to the change in age. So we're really looking for that slope beta. As a first guess, we can compute the average change in the population, called beta bar. Now perhaps our new subject has a genotype similar to another subject. Then we might learn from their phenotype trajectory. Also, other subjects might have similar clinical information or a similar baseline phenotype value, and we learn from their phenotype trajectories in an additive linear fashion. Now these age functions, which we model as Gaussian processes, are kernel-based estimations of how informative other subjects are for this phenotype and this subject. They use similarities based on external data to reevaluate our new subject's phenotype trajectory. Now we evaluated this method on the longitudinal ABNI cohort, and here we see predictions in a few example subjects. For all of these, the algorithm only sees the initial baseline measurement. In purple, we have the actual true observations that are hidden from our algorithm, and in orange is a prediction of no phenotype change. In green is the predicted trajectory using just the average population trend, and in blue is our full model. And we see that when there is only very little change, all these methods predict similarly well. But for subjects with significant changes in phenotype, our model is able to much better predict these diverse trajectories. More quantitatively, and using the same colors, we see that the genetic and clinical information really helps our model here in blue have significantly less relative error than other predictors over the entire population. Now we'll use this scalar prediction combined with an anatomical model to predict entire brain MRIs. We represent the subject MRI as a deformation from an atlas. So at every location we have some displacement that tells us how that voxel should move to form the new subject image. So we have a displacement field from the atlas to the baseline scan, and from the baseline a small displacement field leads to a follow-up later scan of the same subject. Combining these two warps gives us a displacement all the way from the atlas to the follow-up scan. Therefore, in the common atlas space, at each voxel we have these displacements that we can think of as individual phenotypes. And using our model to predict scalar numbers, we can now predict these individual displacements using external genetic and clinical information. And therefore, we can predict an entire displacement field and thus an entire follow-up image. So here is a predicted follow-up scan for a subject with non-trivial temporal changes, and the images look quite plausible. But more quantitatively, we showed the Dice coefficient comparing structures from actual follow-up images to baseline structures propagated through predicted warps. As a guideline in red, we directly registered the baselines to the follow-up scans through the atlas. In some sense, this is as well as we can expect to do. Using the same colors as before, we can see that using genetic and clinical information can clearly help with the anatomical predictions. This opens the door to a wide range of new analysis. For example, let's say that we get a patient with Alzheimer's at their first scan. We can train our model on just healthy subjects and predict what this patient's follow-up scans would look like if they were healthy. Here we can see a predicted healthy image, and the overlay shows significant differences from the real follow-ups. We see that the ventricles would expand significantly less if the patient was actually healthy. So our model for predicting follow-up MRI scans using genetic and clinical information opens up new analysis avenues. And ultimately, we want to answer the more general question of predicting anatomical MRI images without even a baseline scan.